Good afternoon, students. Today we are going to start with the fourth chapter, molecular basis of inheritance. So before starting with molecular basis of inheritance, let's talk about the discovery of the DNA. As we are going to study in this chapter basically the DNA and RNA as a genetic material, right? So there was one of a scientist in 1969, that is Friedrich Miescher, right? So Friedrich Miescher, he was the first scientist who generally worked upon the pus cell, right? Okay, so you know very well the WBC. WBCs are nothing but the white blood cell, which are a major component of the pus cell. Okay, so he generally collected the pus from the bandages which were generally found in the local hospitals, right? Now he generally used a salt solution in order to clean the pus from the bandages. Now when he added a weak alkaline solution to the pus cell got leached up and there was a separation of nuclein which was generally separated in the solution. Now this nuclein is a chemical component which is generally made up of DNA and RNA. So here we are going to study about how the DNA generally came into an existence. So the first experiment was Griffith's experiment, right? Okay, so in 1928, Frederick Griffith was the first scientist. He was a British scientist who for the very first time generally conducted an experiment on a bacteria that generally causes pneumonia. And the name of the bacteria was Streptococcus pneumoniae. It is a type of a bacteria that generally causes pneumonia. Okay, so now when he generally started his experiment, he experimented it on a mice, right? Okay, now he took two different strains of bacteria, right? One was called as the smooth type of bacteria or the S strain, and the second one was rough type of a bacteria also called as R type, right? So first one is smooth type and second one is rough type. Now smooth type of a bacteria was virulent, pathogenic and encapsulated, right? Okay, now what do you mean by your smooth type of bacteria? It was S type, that is it was virulent, it was showing the property of the virus, correct? Okay, that is virulency property. Pathogenic means it was disease causing. It means it generally causes pneumonia, right? Okay, and second type of a bacteria was rough type of a bacteria, which is R type. It was non-virulent and it was non-pathogenic. Non-virulent means it was not going to cause any virulency property, and non-pathogenic means it is not disease causing. It was not pneumonia causing. Now, these two strains of bacteria, S type and R type of bacteria, he generally conducted for the experiment. The observation says that the first observation, when R type of bacteria was injected into the mice, right? Okay, when R type of bacteria, right, when it was injected into the mice generally did not develop pneumonia and mice was survived. Why R type of bacteria, when it was injected into the mice survived? Because R type is non-virulent and non-pathogenic, and that's why the mice was alive and it was survived. Now second observation, he took S type of a bacteria and injected into the mice. When S type of bacteria, which was virulent and pathogenic, was injected into the mice developed pneumonia and finally died. Now why the mice developed pneumonia? Because S type of bacteria was virulent and pathogenic. Now third type, right? Third observation was he took heat killed S type of bacteria, right? Heat killed S type of bacteria means the S type of a bacteria was heated up, and that heat killed S type of bacteria was now injected into the mice, and the observation says that mice did not develop pneumonia and finally was alive. Now why this mice did not develop pneumonia? Because when heat killed S type of bacteria was injected, the S type of bacteria, right, the S type of bacteria, whatever had a pathogenicity, that was been destroyed due to the heating, and when it was been transferred or injected into the mice, finally the mice did not develop pneumonia and was alive. Now the fourth observation, what he used he generally used a very important type of a suggestion, that is when heat killed S type of a bacteria was generally mixed with R type of bacteria. Now he generally used a trick, that is heat killed S type of bacteria. He generally mixed it, he generally fused with R type of a bacteria, and then he injected into the mice. And when heat killed S type of bacteria mixed with R type of bacteria was injected into the mice, mice generally developed pneumonia and finally died. Right? Now, why did the mice develop pneumonia and died? Because heat killed S type of bacteria was mixed with R type, and thus he generally gave a conclusion that the R type, which was non-virulent and non-pathogenic, right? Okay, the R type, which was non-virulent and non-pathogenic, was converted to S type, which was virulent and pathogenic. That is, the harmless strain of R type was generally converted into deadly S strain type of a bacteria, and this change was generally genetic. And thus he said that the R type, which was non-virulent and non-pathogenic, was converted or transformed into the deadly S strain, which was virulent and pathogenic, due to the DNA which was present. And thus Griffith concluded that DNA is a transforming principle. This is generally called Frederick Griffith experiment conducted on a bacteria that causes pneumonia, and the name of the bacteria was Streptococcus pneumoniae. Right? Streptococcus pneumoniae is the name of the bacteria which generally causes the pneumonia. So he took two strains of bacteria, S strain and R strain. S strain was virulent and pathogenic. R strain was non-virulent and non-pathogenic. Okay. Now, second experiment is Hershey and Chase experiment. Very important. Hershey and Chase generally conducted an experiment on a bacteria, right, or on a viruses that generally infects the bacterial cell. So they generally conducted an experiment on the viruses that generally infects the bacterial cell, and any kind of a virus which generally infect the bacterial cell is generally called bacteriophages, right? So he generally took two different types of a medium. Medium 1 containing radioactive phosphorus, that is P32, and medium 2 containing sulfur, that is radioactive sulfur, S35. Right? So two different mediums were taken. Medium 1 containing your radioactive phosphorus P32, and other medium containing radioactive sulfur, that is S35. Right? Now the viruses were generally grown into this two medium, that is phosphorus medium and sulfur medium. Those viruses which were generally grown in the phosphorus medium contain radioactive DNA but not radioactive protein. Why it contains radioactive DNA? Because DNA contains your phosphate or the phosphorus, correct? Okay, so the viruses which were grown in the radioactive phosphorus medium contains radioactive DNA but not radioactive protein, because DNA is generally made up of phosphorus. Whereas the viruses which were grown in radioactive sulfur medium contains radioactive proteins but not radioactive DNA. Why radioactive proteins are present? Because proteins are generally made up of sulfur, and DNA do not contains your what? DNA do not contains your sulfur. Right? Okay. So viruses grown in the phosphorus medium contain radioactive phosphorus but not radioactive protein. And viruses grown in the sulfur medium contains radioactive protein but not radioactive DNA. Why radioactive phosphorus is present? Because DNA is made up of phosphorus. Why radioactive proteins are present? Because proteins are made up of sulfur. Now what did he do is that he generally grew this viruses, or he generally transferred this viruses into the bacterial cell called as E. coli cell, Escherichia coli cell. Right? Now when these viruses were generally transferred into the E. coli cell, the outer protein coat, correct? The outer protein coat was generally removed, correct? Okay, the outer protein coat, that is the capsid, was generally removed by the centrifugation technique. Now the viruses were generally transferred into the E. coli cell or the bacterial cell, right? It was been seen that the radioactive DNA present in the radioactive phosphorus medium had an ability that it penetrated inside the bacterial cell or the Escherichia coli cell, right? But the radioactive protein which was generally present in the radioactive sulfur medium right, did not penetrated inside the bacterial cell, right? So as this radioactive DNA, which was present in the radioactive phosphorus medium, had an ability to penetrate inside the bacterial cell or the E. coli cell, finally Hershey and Chase concluded that as the DNA has an ability to enter inside the bacterial cell, DNA is generally considered as a genetic material. This was the conclusion given by the scientists Hershey and Chase in his experiment. That is called Hershey and Chase experiment. So this experiment was generally based upon viruses that infects the bacterial cell, and they were generally called as bacteriophages. Two different mediums were taken, one medium containing radioactive phosphorus and second containing radioactive sulfur. The viruses were grown in both the medium. Radioactive phosphorus generally medium contains radioactive DNA but no radioactive protein. Sulfur contains your radioactive protein but no radioactive DNA, as the DNA is made up of your phosphorus and proteins are made up of sulfur. Now the protein coat that is captured was when removed by the centrifugation technique. They have been further transferred into the bacterial cell, and the radioactive DNA had an ability to penetrate inside the bacterial cell, whereas the radioactive protein did not. That's why radioactive DNA is generally considered as a genetic material. So these are the two experiments conducted by the scientists. One is Frederick Griffith and second Hershey and Chase, to generally identify, to generally prove that DNA is a genetic material in the living organism. Thank you.